Hey everybody. So last week we were looking at fairly small scale ideas and very modern ideas. So we were looking at um, allele frequencies, how mutations arise and give us new alleles in a population, and then how those frequencies can change, whether they be random or due to natural selection of the allele having an advantage effect on the individuals that have it. Um, and we're now moving past those ideas, but still retaining what we understand about natural selection and how that drives evolution, and taking a step back and looking at those bigger evolutionary ideas. So, on a bigger scale, in terms of the history of life on Earth, how how biodiversity has changed over time, and the evidence that we have for that. So this year's a little different in that we are taking large chunks of content knowledge out of Unit 4, and here's where that fun sort of begins. So looking first at the key knowledge for this section of the textbook, you can see the crossed out sections are what we're leaving out. And in this PowerPoint, I have left the slides in that cover that content, but put that cross through them just to clearly show you what's being left out and what you can choose to ignore in the textbook. So we would normally have a big focus on that timeline of the Earth and how the big events in evolution have taken place and when they happened and how they happened. That has been eliminated, and we're instead reducing it back to the evolution evidence that we have. So for chapter 10, what you really need to focus on is just 10.2. So looking at the evidence of biological change over time, including from paleontology, the fossil record, the relative and absolute dating of fossils, and the steps in fossilization, biogeography, developmental biology, and structural morphology. Those last probably three, they have a few different names that they go by. So that's one of the things that you'll pick up during this conversation is equivalating the word biogeography with talking about distribution biology. So there's a few names that these ones go by, but it all means the same thing. Okay, so get straight into it. So we can ignore this stuff about the geological time scale. You can applaud here because that's quite a large chunk of knowledge that you don't have to worry about this year, so celebrate a little bit. But it shouldn't have too much of a bearing on your understanding of the evidence that we still have for these changes and how they took place. One idea that you should be aware of already, and that is important going forward, is that we did evolve from a single point. So life arose on Earth once as a single event, and from that point, it has diversified, and speciation event after speciation event has taken place. But it would be possible to draw a phylogenetic tree, and show the relationships for every living thing on Earth because we are all related somehow. And we'll talk more about that as we go into chapter 11 as well. All right, so here we sit. We want to know what the evidence for evolution is, and some of these you are familiar with already. Fossils is a big one. So most of the talking that I'm going to do in this PowerPoint is about this first dot point here, fossils. The types of fossil that we know, the process of fossilization, how it occurs, the ideal conditions for it to happen, and how we, after the fact, how we work out how old those fossils are, how long were they in the ground for, how long ago did this organism exist, and how accurate those dating methods are. The rest of them we'll touch on, but it's more, it's not so much content knowledge goes into the remaining three for this one. This last dot point here is what we'll focus on next week. So we'll come to those soon. Okay, so fossils. Fossils are preserved remains, impressions, or traces of organisms found in rocks of different ages. It is nice to have a bit of a definition to go forward with. Fossils are rarely formed. That's a really important idea. We have lots of fossils. There's an endless number of fossils being discovered and have been discovered. But they are not commonly made. Conditions have to be perfect for the fossilization process to take place. Because of that, given an organism's environment at the time of death, there's organisms, there's species that we will never know about because they didn't fossilize. Or their fossils have been destroyed. It is such a rare process that what we call the fossil record, which I'm not sure if I do touch on that on any of these slides, but just in a normal conversation in a class, it would come up. The fossil record is just the record of every fossil that has ever been found. And the most important thing to understand about the fossil record and how fossils relate to each other is that fossils, the fossil record is incomplete. There's gaps in the fossil record. It would ideally show the slow change from one form to another over time, but we just get these small snapshots in time of how organisms have been represented during that evolutionary period. But it's not complete, so the fossil record is ever changing as new things get discovered. But sometimes we're missing that transition fossil between one species to another when we know that they must have some kind of evolutionary link. One has come from the other, but we don't find the in-between form. Sometimes we do. So there's a few examples where it was greatly celebrated in the scientific world that the transition form had been found. So Archaeopteryx is a really good example of that. As part of your style lesson for this week, you're being asked to explore a sort of like an online museum of fossils. So there's a few fossils that you can click on and rotate them and have a good look at their structure. And you're told a little bit of a backstory about them. One of them is Archaeopteryx, so that's a good one to have a look at and perhaps investigate. But it's the transition fossil between dinosaurs and birds. It was the Holy Grail dinosaur fossil that was found with feathers, which really, really changed our understanding of what dinosaurs looked like and how they changed. And ruined the T-Rex for some. Okay, so rarely formed and often only hard parts. So we don't have to worry so much anymore about those geological times and eras, but I still will refer to it for this reason. We see something very early on in the geological time scale called the Precambrian explosion. And it's a point where, as you're moving up through the fossil record, we've got these rarely formed fossils. There's not a great number of them, and then all of a sudden, we have this explosion of fossils. There's a huge diversity and a huge range of different organisms that seem to come out of nowhere. And when they're dated, they all kind of go to that same time period. And where did they come from? How did they evolve so rapidly? It wasn't that they evolved themselves, that they came from nowhere. It was the evolution of the exoskeleton. So what we're seeing there is the point in time when organisms developed those hard outer body parts that were really suited to fossilization. Before that, as multicellular organisms were just evolving, it was really soft-bodied species that existed. And so that rarely fossilizes. It's very hard to find a fossil of a soft-bodied species. But the bony structures, exoskeletons, hard parts of the organisms, that's what fossilization works best on. So for plants, that would be woody materials, the really tough leaves made from cellulose, spores and pollen also make for the best fossils in the plant world, and for animals, bones, teeth, and shell. All right, so that's what our fossils are. How do they form? Okay, so fossilization process is normally what you would think of as a three-step process. Here it's represented as a five-step process, and that's because what's being shown in this diagram is not just the fossilization process, but fossilization to discovery. So this ending here, this is how the fossil's actually forming. For us to be able to see it though, then the last two steps need to take place. So number one, death of the organism. The organism dies and is buried quickly. So quick burial is necessary to prevent scavengers. We want the organism intact. So the organism dies and is buried very quickly. Decay of soft tissues and burial. So as it's been buried, all that soft tissue, muscle, skin, all of that slowly decomposes and decays away. Step 3, sediment piles on top. It's a bit of a few part process here. So we've got not only the accumulation of sediment in many layers going on, as each layer is built on top, it's placing a huge amount of pressure on the remains underneath. And what that's doing is squeezing out all of the water from in between the material that's taking place here. We're going to get to a minute in to talk about the different types of fossils. So what happens from this point forward depends on the kind of fossil we are dealing with. So if you are being called on to explain what's happening in this fossilization process, here's where you would place a bit of detail about the kind of fossil that is taking place. So organism dies, buried quickly, decay of soft tissues, and then sediment layers build quickly on top, which creates a lot of pressure on the remains underneath. And then if you are called to, then you will talk about the discovery process being erosion, which is a natural process by which these built up sediment layers begin disappearing, being worn away, and that could be through water activity, through wind, but the surface layers are being eroded, and then finally discovery by a paleontologist, somebody who's just lucky walking by. But that's the process that you would need to summarize. Okay, fossil types. This is where in class we would bring in a box full of different fossils for us to have a look at. Unfortunately, the best we can do right now is to have a look at that online museum gallery. So jump on and have an explore of those different models that they have of the fossils on that link. But when you're being asked to classify the fossil, this is what you're looking out for. So fossil types that we will encounter: mineralization or petrification. This is where spaces in wood or bone become filled with minerals. The fossil becomes heavy and the organism's remains may be completely replaced by minerals. So mineralization or petrification results in a fossil that is quite heavy. So if you were to hold the piece of petrified wood that I would pull out of that box and place in your hand, it looks like a piece of wood, and it feels like a rock. It's really dense and really heavy, and it's very cold. So it's a very much a rock that is shaped like wood. And it chemically different to the organism, how the organism was in the first place. So if you were describing the petrification process, you would highlight that the chemicals are being replaced by different minerals over time as part of that fossilization process. Impression is a really easy one to understand, and it's a common rock fossil to find. So an organism that has become trapped in the rock will dissolve away and leave an impression or mold behind. So you can imagine you've got a bowl of sand, and if you press something into the sand and pull that out, what's left behind is the impression, the outline of that shape. So that's the impression or mold kind of fossil. A cast is then the opposite of that. A cast is another material that has filled that mold and can be removed from it, and you've got the shape of the organism then intact. So creating a 3D sculpture of the organism using a different material to fill a mold. So that is a cast. A trace. Trace fossils are interesting. If I think you guys probably were the year level that started going down to Inverloch in year 8 to come and have a look at the fossils down there, or you may have done it on your own anyway. But there's that famous fossil down on the Inverloch rocky shore, and I've got it pictured here. I'm having a bit of trouble working out where the fossil is in this picture, but it's the outline of the dinosaur footprint. So that is a good example of a trace fossil. It is not the organism itself as it existed. It is just a trace, a sign that it existed. So it can be things like footprints, as with the Inverloch dinosaur footprint, claw or teeth marks, nests, eggs, and tunnels. And early human, when we look at human evolution, trace fossils would include tools. So just evidence that that organism existed in this period of time. It can also include fossilized poop too. I always forget the name of fossilized poop. It's coprolite. So nice sample of coprolite would be the name for fossilized poo, and that counts as a trace fossil. And probably the most useful fossil that we can find that's going to tell us the most about the organism that left those remains behind. Hey, Chloe. Is mummified organisms. So sometimes fossils can be reserved with little change. For example, woolly mammoths preserved in permafrost in Siberia, or insects preserved in amber. Permafrost is soil that is frozen, and it's a brilliant preserving environment, and gives us a large number of mummified organisms. Why are mummified organisms the most useful fossil to find? It tells us the most chemically about the organism as it existed. They have even managed to extract small amounts of DNA from this kind of fossilized organism. So really, really useful pieces of information about the past. And then some other examples of mummifying environments: volcanic ash, quicksand, peat bogs, and tar pits. One important thing that I've picked up along the way and is worth noting is this point down here. A fossil is best interpreted as an extinct, distant, or close relative of living organisms. We can never know if it is a direct ancestor. So when we come to talk about phylogenetic trees, we never label something, an organism, as being a direct ancestor of an existing organism. The best we can do is is, call it a close relative. We can't say that something evolved from something. We say that they shared a common ancestor. Now, we've talked about these environments being good mummifying environments, but it's worth mentioning too, for the fossilization process and the collection of sediment leading to a good sample of a fossil, the best environments for this to happen is where sediment is accumulating really quickly. So that would be a nice still waters are the best environment. So inland, there used to be an inland sea, in fact, in the middle of Australia, and we do have a really good number of fossils that get found, and a large number of fossils that are found anywhere are of marine organisms. It is harder and rarer to find fossils for land-dwelling animals. Fossilization takes place quite readily in the marine environment because of that quick accumulation of sediment and the weight of the water and all that sediment on top of it. Not all water environments, though, are ideal for the formation of fossils. It has to be still waters. If you've got a fast flowing river, for example, instead of that sediment settling to the riverbed, to the floor, it's washed away too quickly, and probably carrying with it any dead organisms that would happen to fall into the river. So still waters where sediment can readily accumulate is a much better example of a fossilizing environment. Okay, how do we work out how old these fossils are? So this is a relative versus absolute dating piece of knowledge. So relative dating is more inaccurate. It's an estimation based on the presence of something else close by in the same environment. It is an estimation of a fossil's age. Absolute dating is examples of chemical analysis that can be done to provide not exact, an age range, sometimes quite exact, but often a range of years, a time period when this organism would have existed. Okay, so examples of these. Relative age is established through something called stratigraphy. So stratigraphy is where we're looking at the strata layers of the Earth in a particular or at a particular site and looking at those sediments as they've built up. Fossils are found in sedimentary rock. If you can remember back to your junior science years, you would have looked at the different classifications of rock, and you've got sedimentary, which is the accumulation of sediment, as we're talking about with fossilization. You have igneous, which is the formation of a rock layer from volcanic activity, so it's molten rock that has cooled and hardened, and for that very good reason doesn't contain fossils. Because it was molten rock, so anything that was dead inside that layer would have been burnt up. And metamorphic rock, which is either of those two, that's been buried and put under a great deal of heat and pressure over time. Sometimes, very rarely, you do find fossils in metamorphic rock, but they're often distorted and changed enough that they're not readily recognizable. So sedimentary rock is where we find our good, useful fossils. And in these rock layers, the rock layers we call strata layers, the idea is pretty simple. The oldest strata layer is going to be further down because over time the other layers have built up upon it, and the youngest layer then at the top. That's the ideal. So if we found this fossil here in layer C, we can estimate its age based on how much sediment has built up over time on top of it. And if we were to find another fossil in layer A, we would relatively be able to date its age as older than fossils in layer C. Some problems arise with this technique due to tectonic activity of the Earth. So as the crust, the Earth's crust is moving around and the tectonic plates are interacting with each other, it causes things like buckling and layers sometimes to form over the top of one another and fold. So it's not always as straightforward as the layers are in age order, but you would need to be given that detail if you were going to be asked a question about it. Another problem that is encountered sometimes is if you're comparing two sites, so we've got site 1 and site 2 here. Site 1 has layer C 4 layers down, while site 2 has layer C 3 layers down. We know that these two layers are both C, they're both from the same age at the same time, because of the presence of these fossils. Now we call these index fossils. I think this one could be an ammonite fossil. That's probably a trilobite in the back there. And both are good examples of index fossils. So index fossils are fossils that meet certain criteria. They are common. We find them pretty readily. We know how old they are. They are found in a particular age time. They're found in and can be estimated to a particular. They've been dated, so we know that they existed in this particular age and era. So because we find the same index fossil in the same layer, we know that it's relatively the same age. And chemically too, the soil would have certain aspects that are similar. So we would be able to line these two up as being the same age, despite there not being all layers present. So what's happened to layer F over here? Could be a couple of things. It may not have existed at this site, so it was a deposit that was made at site 1 and not at site 2, or at site 2 it has eroded over time, and so we're left with an exposed layer E here. Index fossils are useful not only to estimate the age of a strata layer, but to also estimate the age of a fossil that's found within that same strata layer. So if we didn't know how old this fossil was, but we did know how old this index fossil is, then we can say relatively date this fossil as being to the same time. So that's index fossils. Okay, absolute dating. So this is where our chemical tests come in. So I'll compare two kinds of absolute dating, and they are both radiometric. So we call these techniques radiometric analysis, radiometric dating. Carbon dating is a specific example of radiometric dating. So radiometric dating is basically measuring how much of a particular isotope is left in a sample. And we're relying on our knowledge of the half-life of that isotope. So quick chemistry revision. Both of these symbols represent carbon. Carbon-12 is, I guess, what you think of as the normal form of carbon. Carbon-14 is a heavier isotope of carbon. And it's different to carbon-12 because it has 2 extra neutrons. Carbon-14 is a radioactive isotope. So being an isotope, it's slightly unstable. It's not crazy unstable for carbon. Other isotopes are examples of more unstable in their form. Carbon will break down over time though. And because we know what carbon-14's half-life is, if we can, oh, sorry, backtrack just a little bit. When an organism is alive, the proportion of carbon-12 to carbon-14 is constant. So we know um, the usual ratio of how much carbon-12 and how much carbon-14 is in an organism. And so after an organism dies, if we can measure because carbon-12 will stay constant, if we can measure the carbon-14 that's remaining, that allows us to then calculate how long ago the organism died. So using this graph, and this is all you would ever be asked to do, is to interpret a graph like this. If we are shown, we need to be told how much carbon-14 is remaining in an organism, and we're shown this data, so 100% of its carbon-14 would be present when the organism died. If we can measure how much carbon-14 is remaining in the organism, we can estimate its age, or sorry, fairly accurately state its age using the calculation, using the graph. So if the fossil has 12.5% of its carbon-14 remaining, we can see that, just following it over, roughly, it must have been decomposing for 17,190 years. And that is 3 half-lives of carbon-14. So a half-life is when half of the sample is deteriorated every time. So after one half-life here, we would have 50%, half the sample remaining. And the other half-life, you're halving that half now, would take us down to 25% remaining. So that's 2 half-lives. And 12.5 would be 3 half-lives. And so we know then the age of the fossil based on how much carbon-14 is there. All right, a couple of tricks to carbon dating. It's very useful because organisms are carbon-based creatures. So you can test the actual fossil itself, test it for how much carbon is remaining in that organism. So we can test the fossil itself. And it's very accurate. Carbon dating is a really accurate form of dating. We can narrow things down to within a range of a year. So that scale is really quite small. So it's very accurate, and you can test the actual fossil itself. So that makes it very useful. This is the big drawback. So as we keep decreasing, because carbon's half-life is quite short, compared to some other isotopes that are used, it's depleting at a rapid rate. So carbon dating, the major limitation of carbon dating is that it only is useful up to 50,000 years. So that's a good number to write down. 50,000 years. If a fossil is estimated to be older than 50,000 years, we're not going to be able to use carbon dating to accurately estimate its age. So this is useful for more recently evolved species such as humans. So very useful tool for looking at human evolution. Not so useful for dinosaurs though. If we had a dinosaur fossil, we're not going to be able to use carbon dating on it because dinosaurs went extinct 65 million years ago. So not a useful tool there. We would instead have to rely on another radiometric technique. So much like carbon-14 half-life data, we know the rate of breakdown of many other isotopes. So some useful ones to commit to memory. Uranium breaking down to lead, that's one that is used by paleontologists. And this is my go-to example one, potassium argon, and I've seen that come up in exams before. Potassium argon dating is another radiometric technique, and both of these can be used to date fossils that are billions of years old. So I think some of these go up to 6 billion years, and the world, the Earth, is only 4 billion years old. So that's well and truly going to cover us for the evolution of life. So any fossil that is found, we can use one of the other radiometric techniques to estimate the age of it. Okay, drawbacks, and you would be given data similar to this, the relative breakdown based on how much of the sample is remaining, and that would equate to a number of years. But this wouldn't be in 10,000s of years, it would be in billions of years, millions of years probably, for those two examples. Drawbacks of this one, so it is good because it's got a bigger range that we can go back with, and would be useful for things that are millions of years old. Drawbacks are it's not as accurate, so the window of estimation that we get might be within hundreds of thousands of years. So an organism existed between this 100,000 of years to this 100,000 of years. So it's less accurate, and you can't test a fossil itself for potassium argon dating, because living things don't have a great deal of these elements present in them, especially uranium. So what these are used on instead is igneous rock. So other radiometric methods, apart from carbon dating, they are used to date igneous rock. And so this technique goes hand in hand with stratigraphy. So it's a little bit hard to classify this one as strictly absolute dating. It is absolute dating because you are absolutely dating how old that layer is, but then you need to look at a relative method and say, well, going back to that diagram here of our strata layers, if I've used a radiometric technique and got the age of layer B that way, then I would be able to say that layer C is younger than the age that I have for layer B here. So that summarizes radiometric methods. Okay, very quickly then, biogeography. So we're looking at the distribution of fossils and current species, modern species on the Earth, with the knowledge that due to continental drift, different landforms existed in different periods of time. So if we look at an example here of the ratite distribution map, so we have our big birds, our large birds, ostriches, emus, cassowaries, kiwi, and all the way down to this one who I can't pronounce is the elephant bird, which is actually an extinct bird, and I think the moa actually is as well. These are birds that are similar, and we know they have an evolutionary link, but they exist on different continents. So how do we explain their evolution other than that they were present, a common ancestor was present in the time that Gondwana existed? And from that point, the species have isolated, and through, this would be an example of allopatric speciation, with the geographical barrier separating the new populations, they've evolved separately to one another. So that is another piece of evidence that's often used for a piece of evidence for evolution. So that's evidence for biogeography, and it's called fossil distribution. Another piece of evidence for evolution is structural morphology, or structural homology, homologous structures. So looking at how alike the physical features of different organisms are. And it can also be called comparative anatomy. So if you hear any of those terms, it's all talking about structural morphology. So sticking to what VCAR put into the study design, because that's likely how they'll refer to it in an exam. It doesn't matter what the outer appearance of this structure is, but when you look at the bone structure hidden beneath, the classic example is the forelimb of different mammals. So human compared to cat, compared to whale, compared to bat. Classic example, the wing and the flipper and the arm, they all look on the outside very different to one another, but when you look at the structure hidden beneath, you can't deny the similarities between them. So the arrangement of those bones. And the only way to explain why they're so similar is because it was present in a common ancestor. So before all these species diverged away from each other in their evolutionary history, that structure would have been present, and it has grown and changed with the organism over time. So we've got two examples of structures that can be observed. It is considered homologous if the structure of the physical structure of itself is similar, but the feature the function can vary. So an arm used to grab and move, compared to a flipper used to swim, compared to a wing used to fly. Different functions, same structure. So that would be called a homologous structure. Analogous structures are kind of the opposite. So they have a similar function, but different structure. So where a shark, a penguin, and a dolphin, their limbs look similar on the outside, but the structure hidden beneath is quite different. So this is more, this is an independent evolution of a feature. So these features have arisen independently after these animals have diverged from one another. The structure wasn't present in a common ancestor. They've evolved independently, and so they have that different structure, but the same function. That would be considered an analogous structure. Developmental biology, often referred to as embryology, comparative embryology, but it is essentially the same as structural morphology, but looking at features that are present in an organism before birth. So looking at this as evidence for evolution, the fact that humans and birds and reptiles have developing gill slits as they are evolving. So in the embryonic form, these non-water dwelling organisms at a certain point begin developing gill slits. In humans, it eventually becomes redundant and forms a different structure in the throat, but it is present in the embryonic form of these organisms. So how do we explain that these organisms are growing gills? The genes are still there and active to a certain point during development. The fish obviously would continue growing that feature, but those genes get switched off eventually in all of these ones. But the gene is present and is there because it existed in a common ancestor. So gill slits, and another good example is the tailbone, which again begins developing and then ceases development before the baby is born. I think that's it for chapter 10. So I'll just quickly backtrack. Okay, so we've looked at fossils, the different types, the processes, and how we date them, what biogeography is, what structural morphology is, and what developmental biology is, and how all of these are considered evidence that evolution is a thing. Next week, similar sort of idea. But we're looking at something a little bit more concrete and something that we can calculate and use to determine how alike two species are through DNA and protein comparisons. So that's what we're going to focus on for Chapter 11 next week. But just for this week, looking at our evidences for evolution. Enjoy!